Hello students, the topic for today is gluconeogenesis. Let's start to remind what was glycolysis before that. Glycolysis was process from what to what? From glucose to pyruvate, it's glycolysis. From pyruvate to glucose, this is called gluconeogenesis. When does it occur, gluconeogenesis? It occurs during fasting more than 24 hours and or untreated diabetes. Untreated diabetes is so-called starvation state because there is many glucose in the blood but no in the cells. That's why it's so-called starvation for the cells. Which tissues rely on glucose? They cannot efficiently use fats. They require continuous supply of glucose, and glucose is their primary energy source. These tissues are first the brain, the neurons. They need for energy glucose. Only if there is prolonged fasting periods, they can use some other sources like ketone bodies. But the main source is glucose for the brain. The second ones are red blood cells or erythrocytes because they don't have any choices. They don't have mitochondria. That's why they rely on only glucose molecules with anaerobic glycolysis to lactate. Renal medulla, also eye lenses and cornea, conocytes, testes and working muscles. The next question is, how is the blood sugar level maintained? Actually, students, daily we rely on food, the carbohydrate food, which provides direct glucose after meal. But 2 hours later, the glucose, after the initial increasing spike, peak, it need to be normalized 2 hours later. And after that, if we continue this fasting period, 3 hours, 4 hours and more, without any carbohydrate food, the second source of glucose will be liver glycogenolysis. Maintains blood glucose during fasting for 10 to 18 hours. For example, night sun, during the night sun, we need to maintain the glucose thanks to the liver glycogenolysis. But if there is more than 18 hours with starvation, with no any glucose as a carbohydrate meal, the third process is called gluconeogenesis. Starts during prolonged fasting after glycogen is depleted. Liver glycogen, is depleted. And the sources are non-carbohydrate sources. These are lactate, pyruvate, glycerol, propionate, and glucogenic amino acids. The organelles in the cells are mitochondria, cytosol, and endoplasmic reticulum. What about the organs? Mainly, students, gluconeogenesis occurs in the liver. Remember, liver. This is the organ of gluconeogenesis. 90% is in the liver. And the next, renal cortex. You understood that renal medulla uses glucose with glycolysis, but renal cortex is very similar in the metabolic direction with the liver, which the glycolysis here is around 10%, and the amount is very small in the small intestines. They can use gluconeogenesis also, but usually 1 to 5% totally. But it can increase during fasting or on a diet high in propionate or other short-chain fatty acids. This is the glycolysis process actually, but we need to remind the glycolytic enzymes because we said that glycolysis means from glucose to pyruvate in the end. But the gluconeogenesis means the opposite direction, the opposite reactions. From pyruvate back to glucose in the end. Gluconeogenesis means that we can synthesize our own glucose alone. But if there is no any liver glycogen, only in this situation, and you know that in the glycolysis there are 3 irreversible reactions. These are number 1, number 3, and number 10. These 3 steps, because they are irreversible, need to be bypassed, need to be reversed with specific gluconeogenesis enzymes, gluconeogenic enzymes. And which are the enzymes? We'll see. But all other enzymes are absolutely the same. Look, students, anulase reaction reversible. Number 8 reversible. Number 7 reversible. The enzymes are the same. But let's see what are the names of these enzymes that need to bypass reaction number 1, 3, and 10. Actually, students, the most important and difficult reaction to be bypassed is number 10. Is pyruvate kinase reaction, because it's very difficult to reverse pyruvate to phosphoenolpyruvate. That's why you'll see how, what will be the mechanism. Okay, let's see the glycolytic irreversible enzymes. The first one, hexokinase or glucokinase in the liver and beta cells. They catalyze the reaction from glucose to glucose 6-phosphate, and the opposite enzyme from gluconeogenesis is called glucose 6-phosphatase. Location endoplasmic reticulum, if you're talking about the organelles. But if we're talking about the organs, the only organs with this enzyme are liver mainly, kidneys with 10%, and small intestines 1% approximately. Or, to summarize, the location of this enzyme is in these places that there is gluconeogenesis. But also, this enzyme is used in the end of the glycogenolysis also in the liver, and that's why only the liver glycogenolysis is used for the maintain the glucose in the blood, but not muscle glycogenolysis. Reason: this enzyme is located only in the liver, but not in the muscle tissue. The second enzyme, irreversible in glycolysis, is the main one, called phosphofructokinase 1. It catalyzes the reaction from fructose 6-phosphate to fructose 1,6-bisphosphate. And the corresponding enzyme from gluconeogenesis, which is used for the bypassing of this step, is called fructose 1,6-bisphosphatase. This enzyme is the main regulatory enzyme in gluconeogenesis also, absolutely similar to the same step in glycolysis. Location, cytosol and endoplasmic reticulum. And the last enzyme, which was number 10 in the scheme, is called pyruvate kinase enzyme. Absolutely irreversible reaction again, from phosphoenolpyruvate to pyruvate. This reaction is bypassed by using of all these enzymes: pyruvate carboxylase, phosphoenolpyruvate carboxykinase, malate dehydrogenase, aspartate aminotransferase, and they are involved in one mechanism which is called shuttle of gluconeogenesis. Mitochondria and cytosol are the main localization. And let's see the so-called shuttle and the whole process of gluconeogenesis. Actually, the so-called shuttle, you know from the Krebs cycle lecture, because we talked about that. Actually, what is the object, the purpose? The purpose of this reaction is to bypass the reaction from phosphoenolpyruvate to pyruvate. This is the role. But for that reason, pyruvate enters the mitochondria, then undergoes carboxylation with biotin as a prosthetic group together with ATP, which is hydrolyzed to ADP plus P. Enzyme name is pyruvate carboxylase, well-known enzyme. It is the example of ligase reaction. After that, the product oxaloacetate has three possibilities. Number one, by malate dehydrogenase and NADH as cofactor, undergoes the process of reduction, and oxaloacetate is converted to malate. Malate goes out. Second possibility, oxaloacetate to aspartate. The enzyme is aspartate aminotransferase. The type of the reaction is transaminase reaction, and two substrates are used, not only oxaloacetate, but plus glutamate. The products are two, again, like two substrates. These are aspartate and alpha-ketoglutarate. Aspartate goes out the mitochondria. And the third possibility is the enzyme phosphoenolpyruvate carboxykinase, which uses GTP as energy source to produce GDP, decarboxylation there is, and the formation of high energy bond in the phosphoenolpyruvate. We're ready. Phosphoenolpyruvate is ready, but need to be out in the cytosol. That's why it goes out the cytosol, and in the cytosol can be produced not only by this way, but also thanks to these two enzymes, malate dehydrogenase, but cytosolic enzyme, and phosphoenolpyruvate carboxykinase, the same enzyme, but again cytosolic enzyme. And this, we are ready with the bypassing of the reaction number 10 from glycolysis. After that, all enzymes are the same because they are reversible. Let's see the enzyme names. Phosphoenolpyruvate to 2-phosphoglycerate, enolase reaction, reversible. The next, 3-phosphoglycerate by phosphoglycerate mutase, reversible. The next product, 1,3-bisphosphoglycerate, phosphoglycerate kinase enzyme, reversible. Then, glyceraldehyde 3-phosphate by glyceraldehyde 3-phosphate dehydrogenase, reversible, and etc. to the reaction number 3, which is irreversible. That's why we need specific enzyme of gluconeogenesis, and not only, this is the main one, called fructose 1,6-biphosphatase. This is the main enzyme. And fructose 6-phosphate is produced. Then, isomerase reaction, reversible. And the last, we need the last enzyme, glucose 6-phosphatase, which is used for the degradation of glucose 6-phosphate to glucose, and this is the last reaction which is used for the bypassing the first enzyme of glycolysis, hexokinase. And we are ready. Glucose is produced. Something about the pyruvate carboxylase reaction. Actually, this is the biotin enzyme, or vitamin H, or B7, the other name. This vitamin is used as a prosthetic group, and there is connection with amino group of the lysine residue of the protein part of the enzyme to form so-called biotin enzyme. And after the contact with carbon dioxide is formed the next form of enzyme called carboxybiotin enzyme. And after that start the reaction with transfer of this carboxyl group to the substrate, which is pyruvate, to produce the product called oxaloacetate. All phosphatases, students, are located in the membranes of the endoplasmic reticulum, all of them. These are fructose 1,6-biphosphatase and glucose 6-phosphatase. Both of the phosphatases are located in the endoplasmic reticulum. In this scheme, you can see that in these membranes, there is not only location of this enzyme, but also there is location of some transporters, because we need to be transport glucose 6-phosphate as substrate of the reaction. After that, the products, inorganic phosphate and free glucose, need to be exported out of the endoplasmic reticulum to the cytosol. That's why we need specific transporters. and regulation. How gluconeogenesis is regulated? The first regulation, the first control is hormonal control with glucagon. This is the organ, this is the hormone which activates gluconeogenesis. The action of glucagon is the liver. This is the place of action, liver. No glucogenic receptors in the muscles, they are only in the liver. And activates glucose 6-phosphatase and fructose 1,6-bisphosphatase enzymes. Inhibits PFK1 and pyruvate kinase, which means inhibits glycolysis. One more, gluconeogenesis and glycolysis need to be in balance because they are opposite processes. If there is active glycolysis, GNG will be inactive and opposite. You see that all activators of glycolysis are inhibitors of GNG and opposite. What about the substrates availability? Of course, to start process of gluconeogenesis, we need to have enough substrates to start this process. These are amino acids, glucogenic amino acids, lactate, bicorecycle, glycerol, and etc. And let's see the action, directly the scheme. You know this scheme is a well-known scheme from the previous lecture of glycolysis process. It is the same scheme. Let's see the main activator of glycolysis. This compound, students, is called fructose 2,6-biphosphate. This compound is used for the allosteric activation of phosphofructokinase 1. This is the main activator plus AMP. The inhibitors are ATP, citrate, and hydrogen ions, or NADH. But the same compounds act with opposite direction in gluconeogenesis. Here, I would like also only to remind the formation of this compound, which is the main activator of glycolysis, but the main inhibitor of gluconeogenesis, which is produced thanks to PFK2, phosphofructokinase 2, which is activated by reversible covalent modification. Active form is dephosphorylated form, which means if there is no this compound or very low quantity, no activation of glycolysis, which means inhibition, but GNG will become active. This is the allosteric regulation of both of the pathways. Above is gluconeogenesis. These are the inhibitors, as we said about fructose 2,6-biphosphate and AMP. Actually, students, ADP inhibits both of the enzymes, but the only activator is acetyl coenzyme A. And that's all about the allosteric regulation of gluconeogenesis. Very important is the information about the substrates. Actually, all these substrates are used for the maintain the glucose in the blood, because the object of all this is to produce glucose when there is no liver glycogen. That's why we need all these substrates for gluconeogenesis. Let's see one by one their action. The definition of gluconeogenesis is to form glucose from non-carbohydrate sources. This is the definition. Of course, galactose and fructose can be sources also. Yes, they can be. They could be converted to glucose in the end, but they are carbohydrates, not non-carbohydrate sources. But these first 5 sources, lactate, glucogenic amino acids, Krebs cycle metabolite, glycerol, and propionate, let's see one by one. First, propionate. How is produced propionate? Propionate, or propionate coenzyme A, student, is the end product, one of the end products of breakdown or beta oxidation of fatty acids with odd number of carbon atoms. And this compound consists of 3 carbon atoms and can be converted to methylmalonyl coenzyme A D, then L-methylmalonyl coenzyme A, then succinyl coenzyme A, and then because succinyl coenzymase intermediate of Krebs cycle can enter the Krebs cycle and continues the reaction to oxaloacetate. If oxaloacetate is produced, directly to GNG will be in the end. That's why, if we're talking about the fatty acids and the adipose tissue as a possible source for gluconeogenesis, propionate only, not all fatty acids. Fatty acid with even number of carbon atoms cannot be used for substrate of GNG. Only odd number of carbon atoms and only propionate actually. The small intestines, they use gluconeogenesis thanks to short-chain fatty acids, which are formed thanks to the fermentation of dietary fibers. And these are propionate again, which is a source for GNG locally, but also butyrate with 4 carbon atoms, which is the main energy source of colonocytes, acetate, which is the main short-chain fatty acid in peripheral circulation, but that does not directly participate in gluconeogenesis. The mechanism and physiological role of these substrates like propionate, lactate, amino acids and glycerol is that all these are used for glucose synthesis in enterocytes. Butyrate provides energy for colonocytes and indirectly activates GNG locally. And let's go directly to glucogenic amino acids, which are the main source of gluconeogenesis actually. This scheme shows that any amino acid, glucogenic amino acids, can be redirected to oxaloacetate and then redirection to glucose. But let's see this scheme with so-called glucogenic, ketogenic and mixed amino acids. I will remind you that according to the end products, they differ in 3 types. Glucogenic amino acids are these ones that can be used for glucose. Glucogenic glucose can be produced. These are with green color. For example, alanine, glycine, cysteine, and etc. give pyruvate, or asparagine, aspartate give oxaloacetate, or methionine, threonine, valine give succinyl coenzymase. That's why, students, all these amino acids with green color can be redirected to oxaloacetate, then phosphoenol pyruvate and glucose. But here the question is another question, not only to know which amino acids can be used for gluconeogenesis. The question is where come from these amino acids? And the answer is, these amino acids come from proteolysis, muscle proteolysis, which is need to be activated after the action of cortisol. When there is long-term starvation without liver glycogen, we need gluconeogenesis right now to produce glucose for the main organs, liver, brain and red blood cells. But at the same time, we have increased action of cortisol. It activates proteolysis in the muscles to form all these amino acids which can be redirected to oxaloacetate and then glucose. And all the time you need to think about one more question. If there is so active proteolysis, so active amino acid degradation, what will be produced from all these amino acids out? Because for GNG we need only carbon skeleton. We don't need something from each amino acid. What is this, student? Do you know? What there is in the structure of amino acid that we don't need during the gluconeogenesis process? If you look the structures, you will understand that these are the amino groups. We don't need the amino groups. That's why during the degradation of amino acids, all amino groups will be removed, will be released by processes called deamination. After this deamination, the amino groups will be increased by the form of ammonia in the in the organism, which means the ammonia will be increased. You need to know that when you explain gluconeogenesis, to know that during the gluconeogenesis we have very increased proteolysis to take enough glucogenic amino acids. At the same time, ammonia will be increased, which means at the same time we need specific mechanism for the detoxification of this ammonia. Otherwise, it can be toxic for the brain. And this mechanism for the detoxification of ammonia is called urea cycle. But do you remember one of the connections between urea and Krebs cycle? The connection between urea and Krebs cycle is oxaloacetate again. That's why, remember students, if there is initiation of gluconeogenesis, at the same time there are proteolysis, amino acids breakdown, ammonia increase, urea cycle, and because of the double redirection of oxaloacetate to GNG and urea cycle, the level of oxaloacetate will decrease. And you need to think about what will be happen with Krebs cycle right now. If there is no oxaloacetate, no Krebs cycle. What will be happen with acetyl coenzyme A, which is the second substrate? It cannot enter the Krebs cycle, which means accumulation of acetyl coenzyme A. What will be happen after that? You'll see when we talk about ketone bodies, students. And synergy of organs involved in supplying substrates for gluconeogenesis, corecycled. Means all lactate from the muscles, not only from the red blood cells too, goes to the liver, is accepted in the process of GNG and glucose produced in the end, which again can be exported out of the liver to the blood and can be accepted by the muscles again. Glucose-alanine cycle is the same transfer, but between alanine from the muscles and glucose from the liver to the muscles. In the kidneys, gluconeogenesis requires one important precursor, which is called glutamine. Glutamine, it's an amino acid. The glutamine enters the renal tubuli, and after the deamination once and second time, twice, deamination and formation of ammonia, but ammonia is NH3. After the acceptance of one hydrogen ion, NH4 is produced, which is called ammonium ion. That's why, because of the formation of ammonia, the process is called ammoniogenesis, which means if you're talking about the gluconeogenesis in the kidneys, we need to think about one more process, ammoniogenesis, not only gluconeogenesis. Ammoniogenesis, and one more is used together. It's maintain the isohydria, because this acceptance of hydrogen ions to ammonia helps to maintain the correct pH in the body. That's why three processes occur at the same time in the kidneys. And one clinical case with fructose 1,6-biphosphatase deficiency. Let's see the history. A 9-month-old female infant is admitted to the pediatric clinic urgently after a seizure. The child has been ill for the past few days with a moderately elevated temperature of 38 degrees Celsius. The mother shares for often illness in recent months. The child looked tired and lethargic, did not play as before, did not sleep in the evening, and sweated profusely. The mother associated the beginning of the child's feeding problems with fruit, purees, and juices for the first time. From physical examination and status. During the examination, typical symptoms of hypoglycemia were found: pale, moist skin, decreased muscle tone, and tachycardia. The abdomen in the supine position is above the level of the chest. The liver is enlarged. Imaging studies revealed hepatic steatosis, which means deposition of triacylglycerols in hepatocytes and probable fibrotic changes in the liver. From the laboratory tests, we can see very high leukocytes, hemoglobin is low, glucose is very, very low. The normal reference range is from 3.9 to 6.1 millimole per liter, remember that? Under this range is very severe hypoglycemia, AST, ALT, you know this enzyme? So do you know the full name? Aspartate aminotransferase, alanine aminotransferase, gamma-glutamyl-transferase. All these enzymes are indicators for liver damage. They are very increased in the blood, which means liver damage, and insulin is normal. The child's low blood sugar improved with a glucose infusion. After stopping the infusion, the hypoglycemia recurred and the child's condition worsened again. How can we maintain the normal blood glucose levels? You understood by food, liver glycogenesis and gluconeogenesis. We said in the beginning, thanks to glucagon, because the glucagon is the hormone of starvation and the hormone of glycogenolysis and gluconeogenesis. And let's see the additional laboratory tests and parameters to investigate. They can be biopsy for the liver biopsy to analyze the enzyme activity, and here the result is fructose 1,6-bisphosphatase deficiency. The metabolic consequences will be accumulation of the substrate of the reaction, but let's go directly to the scheme. You'll see after a while a scheme. But before that, the question, why does hypoglycemia occur after feeding on fruit products, after the fruit juice? Because students, there is one first product after fructose metabolism, which is called fructose 1-phosphate. It is metabolized to this compound, but this is the allosteric inhibitor of key enzymes in glycogenolysis, gluconeogenesis, and the result will be hypoglycemia. Let's see the scheme directly. Where is the defective enzyme? This is the defective enzyme, fructose 1,6-bisphosphatase. Students, let's remind, what was the consequences? Tell me the consequences of any enzymopathies. Any. There were only two consequences, students. The consequence number one, no product. Number two, accumulation of very high substrate. These are the consequences only. If the deficiency is here in fructose 1,6-bisphosphatase, means that the product of the reaction, fructose 6-phosphate, will be decreased. But the substrate of the reaction, fructose 1,6-biphosphate, will be increased. And after that, because it inhibits the glycogen phosphorylase reaction, and also after the accumulation of additional quantity of fructose, because of the juices, the fruit juices, fructose 1-phosphate is used for the inhibition of aldolase B from glycolysis and also glycogen phosphorylase from glycogenolysis. That's why the result will be no glucose formation by these processes, no glucose formation because no action of glycogen phosphorylase. That's why the hypoglycemia was so severe hypoglycemia. And therapy will be frequent intake of glucose, exclusion of sucrose and fructose from the diet. Why sucrose? Because it consists of two monosaccharides, one glucose and one fructose, and the result, significant improvement will be. And that's all from today's student.